Hi everyone. Today I would like to talk about genetic mutation in alpha thalassemia. I am Suriana Rahman from Universiti Sains, Malaysia. The content of this lecture will include introduction, molecular basis of alpha thalassemia, laboratory detection, which include hematological methods and DNA analysis, deletional alpha thalassemia, non-deletional alpha thalassemia, and lastly, conclusion. Hemoglobinopathies constitutes the commonest recessive monogenic disorder worldwide. They are caused by variants that affect the genes that direct synthesis of the globin chains of hemoglobin and may result in altered synthesis. For example, in thalassemia syndromes and hereditary persistence of fetal hemoglobin, or structural changes such as sickling of the red blood cells, hemolytic anemia, and polycythemia. Hemoglobinopathies are typically population-specific, with each population having a particular spectrum of thalassemia disease and defective hemoglobin. For the majority of the populations, these variations and their frequencies are typically including a small number of common variants and a slightly greater number of rare variants. In order to increase the detection of the underlying abnormalities in most cases, the diagnostic method may be complemented by patient's ethnic origin and family history, including consanguinity. Worldwide, the most important forms are alpha and beta thalassemia, which affect production of the alpha globin and beta globin chain respectively. Although beta thalassemia is the more clinically significant form, alpha thalassemia occurs at a high frequency across the tropical belt, almost reaching fixation in part of Southern Asia. It has been estimated that about 5% of the population carry an alpha thalassemia variant. Let's go to basic. So the figure illustrates the genes for the alpha chain which are duplicated, and it is located on chromosome 16. On further details, globin genes are organized in two clusters and located on different chromosomes. As I mentioned before, the alpha-like genes are located on chromosome 16 and close to the telomere. The gene for the alpha chain is duplicated, which are HBA2 and HBA1, and the product of the pair alpha genes is identical. Alpha globin genes are expressed consistently at high levels from the beginning of fetal development. Therefore, the effects of alpha globin gene mutations are manifested throughout the fetal and adult life. Generally, the collective of alpha globin genes from the 4 alpha globin genes is estimated to be equal to the total synthesis of beta globin chains derived from the 2 beta globin genes. Now let's learn more about the molecular basis of alpha thalassemia. Alpha thalassemia is caused by loss of numbers of alpha globin genes by deletion or totally or partially inactivated by point mutation. In HBA1 and HBA2 genes and wider locus, more than 120 alpha thalassemia variants have been reported, most of which involve deletion of the alpha globin gene clusters. In addition, more than 1,150 DNA variants causing structural protein variants have been characterized, and some structural protein variants are additionally associated with an altered rate of synthesis. The alpha thalassemia are classified into two main subgroups, which are alpha plus thalassemia, formerly called as alpha thalassemia 2, in which one pair of the genes is deleted or inactivated by a point mutation, and alpha naught thalassemia, which is formerly called alpha thalassemia 1, in which both pairs of alpha globin genes on the same chromosome are deleted. The typical phenotype of alpha thalassemia depends on the number of genes affected, because a normal individual carries 4 functional alpha genes. Individuals who carry a mutation which affects alpha globin genes on a single chromosome associated with mild anemia, and this is called as silent alpha thalassemia if one gene is involved, and alpha thalassemia trait if 2 genes involved. While compound heterozygotes or homozygotes expressing moderate to severe hemolytic anemia are known as HBH disease. And the most severe form of alpha thalassemia is a condition where there is no expression of alpha genes and is called the hemoglobin Bart's hydroxylase syndrome. The excess of beta-like globin chain form non-functional beta chain tetramers called HBH or beta-4 tetramers in adult, while the excess of gamma chain tetramers are called HB Barts in the fetal period. Because of their high oxygen affinity, both tetramers cannot transport oxygen, and in the case of HBH disease, its instability leads to the production of inclusion bodies in the red cells and a variable degree of hemolytic anemia. Therefore, definitive diagnosis of a carrier for alpha thalassemia can only be made by DNA analysis. Now let's go to laboratory detection, which include hematological method and DNA analysis. In general, hemoglobin pattern analysis includes complete blood count, quantitation of HbA2 and HbF, as well as DNA analysis. And DNA analysis of hemoglobinopathies currently in use are based on the polymerase chain reaction. There are many different PCR-based techniques that can be used to detect the globin gene variants. Those in current use for detecting or characterizing nucleotide variations include the amplification refractory mutation system or ARMS, restriction endonuclease PCR or RE-PCR, denaturing gradient gel electrophoresis or DGGE, high-resolution melting analysis or HRMA, Sanger sequencing, pyrosequencing, real-time PCR, reverse dot plot analysis, and microarrays. And the latter two usually as commercially available systems rather than in-house protocol. And for detecting and/or characterizing deletions, Southern blot analysis has been largely replaced by PCR-based methods including CAP-PCR, multiplex ligation-dependent probe amplification or MLPA, and array comparative genome hybridization. DNA analysis has tremendously evolved as compared to the past 40 years. So recently, known and unknown mutation can be diagnosed by using many different PCR-based techniques and DNA sequencing. So, for example, we have dot blot and reverse dot blot techniques, which is allele-specific oligonucleotide probes as hybridization targets for the amplified DNA molecule that complement the oligo probe. This allows direct detection of point mutation or minor deletion that occur in the gene of interest. Next, we have gap PCR. This is currently used to detect common deletion types of alpha thalassemia. It amplifies the DNA sequence by using the primer flanking the deleted region. We also have single-tube multiplex amplification refractory mutation system or ARMS PCR, which can be used for rapid detection of deletion and non-deletion mutation of alpha thalassemia. ARMS technique is also known as allele-specific PCR. MLPA is able to detect large common and uncommon deletion, as well as hemoglobin constant spring for non-deletional mutation. LAM or loop-mediated isothermal amplification has been applied for the rapid detection of several diseases, including thalassemia. This method uses a single-tube amplification with isothermal. The result can be read by the naked eyes based on the colorimetric assay, lateral flow with specific probe, or turbidity. Next, we have Sanger sequencing. And as we know, DNA sequencing analysis is a gold standard method for screening of unknown non-deletional mutations. An advancement in genetic sequencing technology has introduced a new sequencing technique known as next generation sequencing or NGS. This technique has gained capacity to sequence the entire human genome as sequencing capacity increases from several hundred to several thousand base pairs in analysis. Now let's learn about deletional alpha thalassemia. Let's learn about the annotation of alpha plus and alpha naught thalassemia that is used in diagnostic practice. When the expression of a single alpha gene is reduced or absent, this is annotated as alpha plus thalassemia. A patient can be heterozygous or homozygous for alpha plus thalassemia. The allelic expression is reduced either due to a deletion of a single alpha gene, which is deletion type of alpha plus thalassemia, or a point mutation in one of the duplicated alpha globin genes, which is non-deletion type of alpha plus thalassemia. However, alpha plus thalassemia is of little significance to the individual since it is clinically silent. When expressing of both alpha globin genes on one allele is absent, it is called alpha naught thalassemia. As these are usually deletions involving both alpha globin genes, no distinction is made between deletion and non-deletion type, as what we usually use for the alpha plus thalassemia mutations. Compound heterozygous for deletional alpha plus thalassemia and alpha naught thalassemia will show hemoglobin age disease. In normal individuals, alpha globin genes encoding the alpha globin chains are duplicated and localized in a telomeric region of chromosome 16 in a cluster which contain also an embryonic zeta 2 gene encoding for embryonic zeta globin chains, 3 pseudogenes which are pseudo zeta 1, pseudo alpha 1, and pseudo alpha 2, and one gene theta 1 of unknown function. And as we know, we have alpha 2 and alpha 1 gene, which is previously also known as HPA1 and HPA2. This is to show a summary of the most common alpha plus thalassemia deletion and the bars indicating the length of the deletion. This is the most common alpha naught thalassemia deletions and the bars indicate the length of the deletion. To recap again, we have common single gene deletion mutations which are alpha 3.7 and alpha 4.2 and double gene deletion mutations which are alpha 20.5, alpha Southeast Asia, alpha Mediterranean, alpha Thai and alpha Filipino. Now let's go a bit detail about alpha 3.7 deletion. Alpha 3.7 deletion designates a group of 3.7 kilobyte deletions of the 3' end of the alpha 2 gene and the 5' end of the alpha 1 gene with formation of an alpha 2 alpha 1 fusion gene. This results in about 50% reduction in alpha chain synthesis from the affected chromosome. The alpha 1 and alpha 2 fusion gene is downregulated in comparison to the normal alpha 2 gene. Now let's learn a bit detail about alpha 4.2 deletion. It is a 4.2 kilobyte deletion which involves the alpha 2 gene. It is a result in about 50% reduction in alpha chain synthesis from the affected chromosome. Although the alpha 2 gene is usually responsible for about 70% of the alpha chain production, there is some upregulation of the alpha 1 gene when the alpha 2 gene is deleted. Now we go to Southeast Asia deletion. The Southeast Asian deletion alpha naught thalassemia is the most common and severe form of alpha thalassemia found in Southeast Asia and South China. It results from the DNA deletion on chromosome 16, which removes the pseudo alpha 2, pseudo alpha 1, alpha 2 alpha 1, and zeta 1 globin genes. Parents who are both carriers for this alpha naught thalassemia would have a 25% risk of having fetus with homozygous alpha naught thalassemia, causing hemoglobin Bart's hydroxyphthalate syndrome. Although some survivors with hemoglobin Bart's hydroxyphthalate syndrome have been documented, the disease can lead to serious maternal complications during pregnancy, such as hypertension, preeclampsia, polyhydramnios, and severe postpartum hemorrhage. Therefore, it is necessary to provide accurate diagnostic tests for carrier screening and prenatal diagnosis. Okay, let's go about Thai deletion. The Thai type deletion is approximately 33.45 kilobyte in length, which involve both alpha globin genes as well as zeta 2 gene. Detection of these deletions require probes that lie beyond the 58 and 38 deletion breakpoints. In Southeast Asia, the carrier frequency of alpha thalassemia Thai type deletions was 0.2% respectively, depending on the population. Okay, now let's move on to another topic, which is non-deletional alpha thalassemia. Okay, for non-deletional alpha plus thalassemia, nearly 70 types of non-deletional alpha thalassemia have been reported worldwide, and most mutations occur in HPA2 rather than HPA1. And some of these non-deletional type of alpha plus thalassemia may cause a more severe reduction in alpha-globin synthesis as compared to single alpha gene deletions, as there is no upregulation in the alpha-1 gene. A wide range of mutations in the alpha-globin genes have been described, which have an impact on expression, mRNA splicing and degradation, globin chain stability, or on the interaction between alpha chain variant and the alpha hemoglobin stabilizing protein. Other alpha-globin mutations may cause interference with the alpha-beta-globin chain interaction, the formation of the heme pocket, or result in an unstable variant hemoglobin or in a variant with a reduced or increased oxygen affinity. This is example of common non-deletional mutation of alpha thalassemia. So some of these we will go in details. Hemoglobin constant spring is the commonest non-deletional alpha thalassemia. It results from mutation of the stop codon on the alpha-2 gene so that a further 31 amino acids are added to the alpha gene. The mRNA is very unstable and the rate of the alpha chain synthesis reduced to about 1% of normal. Homozygosity for hemoglobin constant spring lead to more anemia than is usual in homozygosity for alpha plus thalassemia, and we also show a different hematological picture. There is usually mild hemolytic anemia, and some cases have mild jaundice and hepatosplenomegaly. The hemolysis is the result of membrane damage by oxidized abnormal alpha chain. Hemoglobin Adana is a form of non-deletional alpha thalassemia mutation located at codon 59 of the HPA2, HPA1 or HPA2 globin gene, leading to glycine to aspartate replacement. These substitutions involve a glycine excess at point of the E helix that is closely attached to the glycine residue of the beta helix. This replacement significantly alters the stability and integrity of the cell's molecule, leading to abnormal precipitates on the red cell membranes, which causes hemolysis and ineffective erythropoiesis. This mutant variant was reported to be linked with a common alpha-1 thalassemia deletion that results in a severe form of HPH disease with anemia. It is characterized by decreased HPA2 level, elevated zeta chain, increased HP bars, and a little of HPH disease. The investigation of hemoglobin Adana is very difficult because the carriers may have normal hematological parameters. Hemoglobin Adana can be detected in the laboratory by alpha-globin gene sequencing or PCR-based amplification refractory mutation system testing. Hemoglobin Quanse is less common type of non-deletional mutation resulting from the HPA2 globin gene, in which amino acid leucine replaces proline, leading to an extended alpha-globin chain. So patients of this condition also experience membrane dysfunction and mild to moderate hemolysis. Hemoglobin Quanse is an unstable hemoglobin variant recorded mostly in southern China and Thailand. It is one of the major alleles that causes non-deletional hemoglobin H in the Chinese population. A novel mutation identified at the initiation codon of HPA2 has led to a conversion of ATG to A to G, and this mutation eliminates the ANCOL restriction site, which may be identified by PCR and southern blot analysis. It results in the reduction of mRNA expression and instability of transcribed mRNA, thus being translated into unstable alpha-globin chains. Specifically, this mutation will reduce the alpha-globin gene transcription by 50%. The mRNA transcribed from the mutated gene will produce non-functional alpha-globin chains during translation. Therefore, this condition will result in hemolytic anemia. The mutation in codon 30 of HBA2 results in codon deletion. The codon that encodes glutamic acid is deleted, hence the alpha-globin proteins will be produced with 140 amino acids instead of 141. So this will lead to the production of unstable alpha-globin chain, which eventually results in RBC detection. And this mutation may not only cause reduction in alpha-globin chain production as it degrades rapidly after translation, but also affects the red blood cell production and lead to ineffective erythropoiesis. This mutation produces a high unstable protein that tends to degrade rapidly after translation, thus reducing the level of functional alpha-globin chain. Now let's learn a bit about hemoglobin evora. The missense mutation of hemoglobin A2 in codon 35 results in the production of proline instead of serine, leading to the production of a variant known as hemoglobin evora. Proline is incapable of participating in the formation of alpha helix protein, which causes a conformational change in alpha-globin chains. And this mutation was reported among patients of Caucasian descent in Portugal. The distribution of this mutation remains unknown as it is rarely reported. And the first case related to this point mutation was reported in 2001, with patients showing mild macrocytosis and was hypochromic. Now we are at the end of the presentation. In conclusion, alpha thalassaemia is a common genetic disorder worldwide, thus require proper care and management. Although delusional alpha thalassaemia is common than non-delusional ones, the latter will result in a more severe and significant clinical presentation. Knowledge on the molecular basis that regulates the expression of the alpha-globin genes is vital for the accurate understanding and differentiation of alpha thalassaemia. Nationwide screening programs, pre- and post-marital screening, including infant screening, have well been developed. Accurate investigation of thalassaemia requires screening of the population and identification of families who are at risk. Examinations of complete blood count for hemoglobin level and red cell indices, peripheral blood film morphology, together with hemoglobin electrophoresis, and most importantly, modern DNA analysis will help for easy differentiation. This is the reference. Thank you very much for listening.